Okay, before we begin, are there any questions? Not hearing anything. My computer's a little slow. Let's open up my syllabus. Let me share my screen.
I have one question. I just wanted to confirm, like the last quiz, what pages chapter three, the end of chapter three, will include for this exam tomorrow?
I'll have to get you that in person. I don't know exactly the page break off, but it's about where we stopped. Let's go to the schedule. Can you see my screen?
Yes.
Thank you. So on chapter three, it's about where the first day stopped. I can get you the exact page number later, but I don't have it off the top of my head, so I'll have to look that up.
Okay, yeah, that'd be super helpful. Thank you.
All right. Could you send me an email just to remind me to look that up? Because after the two lessons today, I'll probably not remember the questions. So if you could send
Yeah,
me,
I sent you one earlier in the day today.
okay, thank you.
Yep.
All right. Today is February 10th. Let me scroll down a little. And we do have a quiz this Wednesday, quiz three, covering lab 5 and 6. So I'm having a hard time reading here. Let me blow that up a little. Chapter 4 and the end of chapter 3. And I did state that it's about where the first day breaks off. And I will get you exact page number after I look it up, but I don't have that memorized. Um, Today we're supposed to be working on chapter 5. I think still on chapter 5, and hopefully we'll actually start chapter 6 a little bit. I don't know if we'll succeed. There is a lab today, lab 7. Any questions about what we're doing? All right, no questions. Let's move on. Chapter 5. I do realize my clock is a little slow and I need to take it into Clark College. It used to be when I hooked up to Clark College, it would synchronize the clock here. So be realizing that my clock is a little slow and I do go by my clock for setting the class. So until I take it into Clark College, I can't change the time. They have that locked down. You have to be an administrator, and I'm not an administrator to change the time. All right, I talked about the generation of ATP last time and stated that it can happen in three ways: by substrate level phosphorylation, by respiration, and then lastly by photophosphorylation. Photophosphorylation only occurs in photosynthetic organisms. Uh, the energy production, it's energy is released from molecules in controlled ways, like when energy is released from the burning uh, glucose. The energy is not released in a single burst, such as when I were to take a cup of glucose and then take a blowtorch to it. The energy would be all released at one time, and that energy would be something like 1,000 degrees. And in our cells, as you know, 1,000 degrees would kill our cells. So we release the energy in stepwise process, and then the cell can remain at close to 37 degrees. And the electrons from a donor molecule like glucose are passed from one compound to another through a series of redox reactions, meaning oxidation reduction reactions. And there you're seeing the oxidation reduction reactions of aerobic respiration. We won't go into each step there, but the point is the electrons from glucose pass to NADH or FADH2. These two electron carriers pass it to these here, and then each one is oxidized and then reduced or is it reduced and then oxidized, whatever. And the electron eventually will pass to oxygen in aerobic respiration. So oxygen is said to be the final electron acceptor. Any question about any of that? And the point of doing it stepwise is so the energy is released in steps, and that way the cell can maintain its temperature of around 37 degrees. Carbohydrate catabolism is used by most organisms as their primary source for obtaining cellular energy. The most common energy source is glucose, but any food item that is digested can be used for cellular energy, meaning we can eat all lipid diet or an all protein diet, and you can burn those to get energy, which will then be stored as ATP. But to produce energy from glucose, microorganisms use two general processes, respiration and fermentation, to make and store their ATP. There are two types of respiration, aerobic respiration and anaerobic respiration. Let's look at how do I open that? It's not giving me the link. There it is. Let's look at a little movie about introducing us to aerobic respiration. After the commercial.
Soon we'll be telling the whole stadium how Liberty Mutual customizes car insurance to save people hundreds. Whoa.
All cells need energy to stay alive. Usually your cells get their energy through the process called aerobic respiration.
Can you
The
guys
word
hear that?
aerobic refers to oxygen. In
Yes.
aerobic respiration, oxygen interacts chemically with a molecule of glucose. The glucose is broken down into carbon dioxide and water. This process releases energy, which the cell stores in the chemical bonds of ATP. The first stage of aerobic respiration, glycolysis, occurs in the cytosol, the jelly-like part of the cytoplasm. Glycolysis breaks each molecule of glucose into two molecules of pyruvate, energizes two electron carrier molecules, and produces two molecules of ATP. The next two stages of aerobic respiration are carried out in the mitochondria. Pyruvate enters a mitochondrion. Once inside, it is modified and enters the Krebs cycle. The reactions of the Krebs cycle break the pyruvate molecules down to carbon dioxide, create two more molecules of ATP, and energize more electron carrier molecules. The final stage of aerobic respiration is electron transport. The electron carrier molecules that were energized in the earlier stages deliver electrons and hydrogen ions to the electron transport chain. As the electrons are passed down the chain, energy is released and used to make ATP. This final stage produces as many as 32 molecules of ATP from each molecule of glucose.
Okay, any questions about that? Let's go ahead and shut that down. So here we're looking at an overview of aerobic respiration and fermentation, comparing the two. Let me see if I can blow that up. Nope, can't reach it. So aerobic respiration happens in three principal stages. Glycolysis, the first stage, the Krebs cycle, the second stage. Oops. I get that to the top now. And then the third stage, the electron transport chain. Essentially, electrons flow from glucose, which comes into glycolysis. There it is. Glucose is an energy-rich molecule. Glucose is converted into CO2 and water. they are energy poor molecules. And then the energy that's given off, a lot of it is used to obtain ATP, and we get a high yield of ATP from aerobic respiration. In fermentation, on the right here, we do have glycolysis, which is the first stage of fermentation. There is no Krebs cycle, there's no electron transport chain. Instead, glucose is converted into pyruvate. Pyruvate will then be converted in the fermentation steps to the fermentation end product, which can be ethanol or lactic. The ATP yield is much lower. Any question about any of that? This is showing you, I think this would be good enough, an overview of aerobic respiration, the 3 steps: glycolysis, the Krebs cycle, also called the citric acid cycle, and then the electron transport chain, which has a whole bunch of names, including oxidative phosphorylation shown here, chemiosmosis. There's also the electron transport chain, the electron transport phosphorylation, a whole bunch of names for the electron transport chain. And if you take a look at this, the pink area would be a cell. So the aerobic respiration happens within a cell. It does start from glucose, which by glycolysis, the first step, will be converted into pyruvate. This is showing you that this step happens in the cytoplasm of the cell, meaning it happens inside the cell membrane, and in a eukaryote, it actually happens outside of the nuclear membrane. It also happens outside of the mitochondrial membrane, and that's why we say it happens in the cytoplasm. Pyruvate is generated. There is some ATP made. There's some NADH also made. We'll talk about NADH in a little bit. The pyruvate then moves into the mitochondria, inside the matrix of the mitochondria, meaning it moves inside the outer membrane of the mitochondria and inside the inner membrane of the mitochondria. And then we have the Krebs cycle happening after the guy who invented it. That's the way I normally call the Krebs cycle, but it does have 2 other names. One is the citric acid cycle, or TCA for the citric acid cycle. And the other one is, gosh, what the heck is it called? Some other acid cycle, and it's abbreviated TCA as well. Don't remember the name of it. Sorry, just slipped my mind. The Krebs cycle, the citric acid cycle, and citrate is the first sugar made in the cycle. So that's why it's called the citric acid cycle. But anyways, that happens inside the matrix. And then a large number of NADH and FADH2 is made by the citric acid cycle or the Krebs cycle, as well as some ATP. This is where the CO2 gets generated as well. And then the NADH and FADH2, including the NADH from glycolysis, carry the electrons to the electron transport chain. The electron transport chain happens across the inner mitochondrial membrane of the mitochondria, and that generates ATP in the electron transport chain, or the 3rd step of the aerobic respiration. Any question about any of that? All right, that's just the overview. And then it's showing you that in a eukaryote cell, the Krebs cycle, as well as the electron transport chain, happens in the mitochondria. The Krebs cycle happens in the matrix of the mitochondria. The electron transport chain happens across the inner mitochondrial membrane. All right, so in aerobic respiration, or actually any form of respiration, glycolysis is the first step. Glycolysis does have a different name. It's also called the Emden-Meyerhof pathway. I don't use that terminology. I might be butchering the names too because they're German and I don't speak German. I just call it glycolysis. Glycolysis is the oxidation of glucose to pyruvic acid. Glycolysis occurs in most living cells. It can occur in the presence or in the absence of oxygen. It does produce some ATP and some NADH. The NADH will take the electrons to the electron transport chain. We'll talk about that later. In glycolysis, shown here, we start with glucose, a 6-carbon molecule, and we end up with 2 pyruvic acid, also called pyruvates, which are 3-carbon molecules. That's why we get 2 of them. Pyruvic acid is the same as pyruvate. It's just pyruvate has the hydrogen removed, and then pyruvic acid has the hydrogen on it. The cell will treat pyruvic acid and pyruvate the same, and you really need a biochemist to note the difference between the two. Any question about any of that? So, glucose is converted by glycolysis into pyruvate acid. In these steps shown here, it's about 10 steps. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And it is true that we make this here, and then that will be converted into that. And then we go down this step again. So you can make that down here if you want, or we go down this step twice. Any question about any of that? You do need 2 ATP to get glycolysis to run, but we get 4 ATP, 2 here and 2 there, out of the reaction. So we say that the net gain of glycolysis is there's a net gain of 2 ATP made by glycolysis. Any question about any of that? Now, when I took undergraduate biochemistry, I needed to know the names of each intermediate, starting with glucose, ending up with pyruvate, and I needed to know the names of all the enzymes in each of the 10 steps, including this one here, so that would be 11 steps, converting that to that. And since I had to know that, I think it's fair that I require you guys to know that. What do you guys say about that?
Terrible idea.
Hmm. You don't need to know all that. You do need to know that
Thank
the
you, thank
reaction
you.
starts with glucose. You get a net gain of 2 ATP from glucose going through glycolysis. You get 2 pyruvates generated from 1 glucose, and you get a net gain of 2 NADH, or I should just say, you get 2 NADH out of the reaction. You do not need to memorize glucose plus 2 ATP plus 4 ADP plus 4 inorganic phosphates plus 2 NADH generate 2 pyruvic acids. You do need to know that one, but 4 ATP, 2 NADH, 2 hydrogen, and then 2 water. You don't need to know that, but you do need to know this. So you start with glucose, and then you end up with 2 pyruvic acid or 2 pyruvates. Like I said, I'm going to use those interchangeably. You generate 2 NADH and a net gain of 2 ATP. Any question about glycolysis? You then have respiration follow glycolysis. There are 2 types of respiration: aerobic respiration, where the final electron acceptor is oxygen, and our cells engage in aerobic respiration, but you can have anaerobic respiration, where the final electron acceptor is a molecule other than oxygen. It can be different molecules depending on what type of anaerobic respiration it is. It could be an iron molecule. It could be A nitrogen compound like nitrate or nitrite. Could be a sulfur compound like sulfate. Could be another compound as well. In respiration, ATP is generated by oxidative phosphorylation, and you get 2 ATP out of aerobic respiration for one glucose molecule. You get 2 ATP for anaerobic respiration as well. We'll talk about anaerobic respiration later. I'm just pointing out that there are two types of respiration. One where the final electron acceptor is oxygen, and another where the final electron acceptor is a molecule other than oxygen. Any question about any of that? All right. The first stage of the Krebs cycle is the preparatory step. In the preparatory step, pyruvic acid, which is generated by glycolysis, has to be changed into it's acetyl coenzyme A. Coenzyme A combines with the breakdown of pyruvic acid is converted into, acetyl coenzyme A. One CO2 comes off, we do generate one NADH, but realize the preparatory step is going to run twice for each molecule of glucose, because one molecule of glucose will generate 2 pyruvic acids, and so the preparatory step will run twice for each molecule of glucose. You'll note that oxygen has not been used yet, and the preparatory step has to occur because pyruvic acid cannot directly enter the Krebs cycle. It has to be converted into acetyl coenzyme A. We then have the Krebs cycle following the preparatory step. And when we're talking about aerobic respiration having 3 steps, we're linking the preparatory step in with the Krebs cycle. The Krebs cycle is also called the tricarboxylic acid cycle, or TCA for tricarboxylic acid, or the citric acid cycle, which is also called TCA for T, C, citric, and then A for acid. It's called the Krebs cycle, and that's the way I use it as well as your textbook most of the time, because this was discovered by Dr. Krebs. It's called the citric acid cycle because the first compound made in the citric acid cycle is citrate, or citric acid. And it's called the tricarboxylic acid cycle sometimes because there's a lot of 3-carbon molecules made in this cycle. Any question about any of that? And for my use, I'll just call it the Krebs cycle. And if you learn that, it'll be good with the textbook too. All right, blow this slide up. So in the Krebs cycle, we first have preparatory step where pyruvic acid is converted into acetyl coenzyme A. NADH is generated, 1 CO2 comes off because acetyl coenzyme A is a 2-carbon molecule, pyruvic acid is a 3-carbon molecule. And that's the first CO2 made from glucose. And then acetyl coenzyme A combines with oxaloacetic acid to make citric acid. Citric acid is made into this compound. We get some NADH and CO2 made, which is then converted into this compound, then converted into that compound. We get some NADH and CO2 made here. This compound is then made into that compound. We generate some ATP here. This compound is then converted into that compound. We generate some FADH2 there. This compound is then converted into that compound. That compound is then converted into oxaloacetic acid. We generate some NADH there. And then we have ended up in the molecule that started the Krebs cycle, oxaloacetic acid. And this is why it's called a cycle, because as long as your cell is alive, the cycle runs, just runs in a circle. That's why it's called a cycle. Any questions about any of that? All right, for each running of the preparatory step in the Krebs cycle, we generate one ATP molecule, shown here. We generate four four NADH molecules, one in the preparatory step, one here, one there, and then one there. So a total of four for one pyruvic acid. We generate one FADH2 right there. We also generate three CO2s, one in the preparatory step right there, one right here, and one right there. And that means all of the carbons in pyruvic acid, three of them, are converted into three CO2s. Realize that for each molecule of glucose, you're going to be having the preparatory step and the Krebs cycle run 2 times, because glycolysis will make 2 pyruvic acids per molecule of glucose. So you have to be careful how the question is worded on a quiz. Will it ask how many ATP are generated per pyruvic acid? The correct answer is 1. But if it says how many ATP are generated per molecule of glucose, the correct answer is 2, because glucose will run the Krebs cycle and the preparatory step 2 times. Any question about any of that? All right, the NADH as well as the H plus and the FADH2 are carrying the electrons to the electron transport chain. And the cycle ends with the molecule that started the cycle, oxaloacetic acid. So as long as acetyl coenzyme A is around in the cell, this cycle will continue. And that's why it's called the cycle. Any question about any of that? All right, so you should note that all of the carbons in glucose end up as CO2, and that's why we breathe CO2 out, because we burn glucose, which is converted into CO2. And that happens in the preparatory step and the Krebs cycle. None of that CO2 that is generated comes from the oxygen we breathe in. Any question about any of that? All right, this is showing you an overview of aerobic respiration, comparing it to fermentation. And after the Krebs cycle, the NADH, the FADH2, not shown here, carries the electrons to the electron transport chain. It's true of the NADH being generated in glycolysis as well, carrying the electrons to the electron transport chain. In the electron transport chain, we then kick ATP production into high gear. We're in aerobic respiration. We generate a lot of ATP. The final electron acceptor is oxygen. And that's why it's called aerobic respiration. Any question about any of that? All right, the electron transport chain, also called the ETC for short, it does have a lot of different names. Chemiosmosis is one. Oxidative phosphorylation is another. You could call it the electron transport phosphorylation as well. Generally, electron transport phosphorylation is easy, similar enough to electron transport chain, but doesn't confuse people too much. I tend to call it the electron transport chain. I think that's the way your book tends to call it as well. I won't call it after this step any further, chemiosmosis or oxidative phosphorylation, but those are terms for the electron transport chain as well. The electron transport chain is a series of carrier molecules that are in turn reduced and oxidized as electrons coming from ultimately glucose are passed down the chain of molecules. And this chain is mostly a chain of proteins in a membrane, and that's why it's called an electron transport chain. And the electrons flow from one member of the chain of members to the next member. Energy is released as the electrons are transferred from a high energy to a lower energy compounds. And this is called chemiosmosis. You can watch the little video if you wish. I'm just going to talk about it. In eukaryotes, the electron transport chain happens in the inner mitochondrial membrane. In prokaryotes, it happens in the cell membrane of the cell because prokaryotes do not have a mitochondria, but we do have an electron transport chain, which is a chain of proteins in a membrane, and it's the cell membrane in a prokaryote cell because they don't have any other membranes. But in a eukaryote cell, it happens in the inner mitochondrial membrane. Any question about any of that? All right, let me blow this up just a little bit. So in the electron transport chain, we have the electrons being carried by NADH or FADH2. They bring the electron to the NADH brings it to the first protein in the chain of proteins. FADH2 actually brings it to the second protein in the chain of proteins. And then the electron moves from one member of the chain of proteins to the next, and then to the next, to the next, to the next. In aerobic respiration, that electron leaves the last chain in the protein, last member of the chain of the protein, and then that electron flows to oxygen. Oxygen combines with 2 electrons and 2 hydrogen ions to form water. And that's why we need to breathe in oxygen, by the way. All the oxygen you breathe in that is used will be converted into water by aerobic respiration. Along the way, as the electrons move through some of the proteins in the chain of proteins in the membrane, some of the electrons give the protein the energy it needs to transfer hydrogen from this side of the membrane to that side of the membrane. And that happens here in the first protein, in the third protein, and then the last protein. As the electron is moving through that protein, it gives the protein the energy it needs to pump hydrogen from this side of the membrane to that side of the membrane. That creates a twofold gradient. We have more hydrogen on this side of the gradient than this side, and we have more positive charges on this side of the gradient than that side. By the laws of thermodynamics, these H plus ions will want to move from this side of the membrane to that side of the membrane. To move through the lipopolysaccharide layer, the bilayer of the membrane, is not possible for the hydrogen ions. They can only move through this protein here. And when they move through this protein, the hydrogen ion gives this protein the energy it needs to phosphorylate ADP and phosphate into ATP so that we generate ATP, one molecule of ATP, for each hydrogen that moves across this protein. And that protein is an ATP synthase protein. Any question about any of that? And how that's happening is that we have a twofold gradient. There's more H plus ion over here, more hydrogen, more plus charges than here. and the hydrogen will want to move to this side of the membrane, and it can only move through this protein. Any question about any of that? All right, where is this membrane? It's the inner mitochondrial membrane of a eukaryote cell, and it's the cell membrane of a prokaryote cell. Any question about any of that? ATP production kicks into high gear in aerobic respiration. In prokaryote cells, you can generate 34 molecules of ATP per molecule of glucose in the electron transport chain. And prokaryote cells are a little more efficient. Why would generate 34? In eukaryote cells, you generate 32 to 34 molecules of ATP per molecule of glucose for the electron transport chain. Specifically, most of the cells in your body can only generate 32 molecules of ATP per molecule of glucose in the electron transport chain. However, the cells of your heart, your kidney, and your liver can generate 34 molecules of ATP per molecule of glucose. Why do you think your heart cells, your liver cells, and your kidney cells are a little more efficient than the rest of your cells in your body, such as your skin cells or your nose cells? Think about your heart. What is it always doing? Think about, I don't know, your arm, the muscle in your arm. What's the difference between the muscle in your heart and the muscle in your arm?
Does it have to do with voluntary and involuntary muscle contraction?
Not exactly. We could say something like the muscle in your intestines, which help you digest your food by moving the food one way to another. That's involuntary. So it's not really that, whether it's voluntary or involuntary. And it's true the heart is involuntary, but like I said, it could be the muscle in your intestines, which is involuntary. It can only generate 32 molecules of ATP, but your heart generates 34. Why is your heart more efficient than the muscle of your intestines, for example, or the cell of your skin? What's your heart always doing? As long as you're alive.
Beating, pumping.
Yeah, it's always working. The heart is always working. It's always pumping as long as you're alive. And your kidney is always recycling the blood, purifying the blood. And the liver is always working as well. It really, what it's doing is checking the blood to make sure there's no toxin in it and removing it. So it's filtering the blood all the time. A little different than the kidney. The kidney is removing waste from the blood. And I suppose the liver is too. It's just removing it and then oxidizing it. But my point is the liver, the kidney, and the heart are always working. And so they're a little more efficient at generating ATP because those cells are always needing ATP. Whereas the cells of your skin or the cells in your muscle in your arm, they're not always working. And so they don't need to generate 34 ATP molecules. Any question about any of that? All right. In aerobic respiration, the final electron acceptor is oxygen. Oxygen combines with the 2 electrons and 2 hydrogen ions to make water. And that is what happens to all the oxygen you breathe. It is converted into water. Some of that water will be breathed out. Some of the water will be used for the blood, used for the organism to provide water for the body. Desert animals are actually very good at obtaining their water needs from aerobic respiration. We're not a desert animal, so we're not as good. But desert animals can generate 60% of the water they need from aerobic respiration. I don't know how much water we generate because we're not a desert animal, but it's a little less than 60%. Any question about any of that? All right, this is showing you the electron transport chain compared to fermentation, which has no electron transport chain. And there's the steps of the electron transport chain. And there's the molecules in the membrane. So there's the steps. And the electron will flow to oxygen or aerobic respiration, which will be converted to water. And here we're seeing the electron transport chain in the membrane of the cell, which in a mitochondria, shown here, it's the inner membrane of the mitochondria, and the electrons flow across the membrane, moving from one electron to another, flowing to oxygen. That gives some of the proteins the energy they need to pump hydrogen ions from this side of the membrane to that side. This will be in the inner mitochondrial membrane shown right there. So the hydrogen ions are pumped in between the inner mitochondrial membrane and the outer mitochondrial membrane. And that's to protect the cell from the hydrogen ions. And that's why we have an outer membrane on the mitochondria, so that we keep the hydrogen ions inside the mitochondria, and then they'll flow through this ATP synthase protein to make ATP. In a prokaryote cell, this is not the inner mitochondrial membrane because prokaryotes do not have a mitochondria. It is the cell membrane. And where are the hydrogen ions flowing? What's right here on a prokaryote cell, right outside the cell membrane? Come on, you guys know this.
A cell wall.
Yeah, that's the cell wall. So the hydrogen ions are actually flowing inside the cell wall. And then they will come back through this protein here. Just to know that the hydrogen ions are pumped into the prokaryote cell wall. Any question about any of that? By the way, the hydrogen ions are not retained by the cell wall. The hydrogen ions can go right through the cell wall, but they stay in the cell wall until they move through the ATP synthase, at least most of the time. Excuse me. We already talked about that. I did also mention that the hydrogen ions are pumped by certain of the proteins in the chain of proteins, the electron transport chain, and they pump hydrogen ions over here. This creates a twofold gradient called the proton motive force. And these protons will want to move to this side of the membrane, and they can only move through this protein in the membrane, which when they move through it, that generates ATP. So we're using the hydrogen ion gradient to make ATP. It's called chemiosmosis, and that's how ATP is made. It's highly efficient, by the way. Let me see if I got that in there. I don't have it here. It is highly efficient, and I believe 40% of the energy that's stored in glucose can be converted to the energy in ATP. And you say, well, 40% isn't very good. That means about 60% is lost. The energy is converted into heat. You might say 40% isn't very good. But if you take a look at the very best engine we've made, humans have made, the best we've ever gotten an engine to convert that energy in whatever we're burning, like gasoline, into the energy of locomotion. our engines can only reach between 20, the very best, maybe up to 25% efficiency. So the very best engine we have that man can make is only 25% energy. The electron transport chain and aerobic respiration can convert 40% of the energy in glucose into the energy in ATP. Much better than any engine that humans can make. Any question about any of that? All right, I did mention that electron transport chain is also called oxidative phosphorylation. It's oxidative because the electrons ultimately will flow to oxygen, and it's called phosphorylation because ADP will become phosphorylated. Phosphorylation just means you're adding phosphate group to it to make ATP. And as I stated, the complete summary reaction for oxidative respiration, in a prokaryote cell, you generate 38 ATP molecules per molecule of glucose. In a eukaryote cell, you generate 36 to 38 ATP, depending on what cell we're talking about. The heart and the liver, the kidneys can generate 38. All the other cells in your body can only generate 36 molecules of ATP per molecule of glucose. You do need to know the summary equation. Glucose C6H12O6 plus 6 oxygen molecules plus 38 ADP and 38 phosphate generate in aerobic respiration 6 CO2. All of these CO2 come from glucose, 6 water molecule. All of those oxygen and water come from the oxygen and oxygen gas, and then the 38 ATP. Any question about any of that? Comparing aerobic respiration, 36 to 38 ATP, compared to fermentation, only 2 ATP. Oh, that's not compared. Sorry. Gotta blow this up so I can see it. This is showing you where we generate the ATP, 32 to 34 in electron transport chain. It's showing for a prokaryote only 34. And then 2 ATP in the Krebs cycle, 2 ATP in glycolysis, for a total of 38 ATP. Any question about any of that? All right, let's talk a little bit about anaerobic respiration. Anaerobic respiration is the same as aerobic respiration. It's just that the final electron acceptor is not oxygen. It's some other molecule other than oxygen. So the electron will leave the electron transport chain and then combine with a molecule other than oxygen, depending on which form of anaerobic respiration it is. It could be iron compound, could be a nitrogen compound, could be a sulfate compound. Anaerobic respiration, glycolysis and the Krebs cycle still occur. There is an electron transport chain as well, but this electron transport chain is a little shorter than the chain of aerobic respiration. So let me go back to this right there. Pull that slide up. It's still not real good, but this is the electron transport chain of aerobic respiration right here. And it's a little shorter, so we generate less ATP in anaerobic respiration to compared to aerobic respiration. And it depends on which form of anaerobic respiration it is. Some of them only miss the very first protein. Others, all they have is the last protein. So it depends on which form of anaerobic respiration it is. The generation of ATP will depend on which form of anaerobic respiration it is, but it's always something greater than 2 and always less than 38 molecules of ATP. So something between 2 and 36. The very best forms of anaerobic respiration only generate 36 molecules of ATP, which, remember, for prokaryotes, the very best form of aerobic respiration, actually the only way prokaryotes, is they generate 38 molecules of ATP, a little better than the best anaerobic respiration. And then the very worst, I think it's down to 4 molecules of ATP per molecule of glucose for anaerobic respiration. Because anaerobes generate less ATP per molecule of glucose, anaerobes tend to grow slower than aerobes. Because if you perform aerobic respiration, that's the best way to generate ATP. You'll get the most ATP from anaerobic respiration. Any question about any of that? This is showing you all forms of respiration do have glycolysis, the preparatory step in the Krebs cycle, and an electron transport chain. It's just their electron transport chain is a little smaller than the electron transport chain in aerobic respiration. Fermentation, all it has is glycolysis and a fermentation step or steps after that. Any question about anaerobic respiration? If not, let's move on to fermentation. We do generate ATP in fermentation, but not much. We only get a net gain of 2 ATP from fermentation, and all of these ATP generated occur in glycolysis, which is always the first step of fermentation. The second step of fermentation is 1 or 2 fermentation reactions, which convert the pyruvate coming out of glycolysis to the fermentation end product. So in fermentation, we generate 2 pyruvic acid molecules. By glycolysis, we generate 2 ATP. We do generate NADH as well, and that's shown here. Glycolysis generates 2 ATP and 2 NADH. That NADH is not saved, though, for aerobic respiration. The NADH is used to convert pyruvic acid to the fermentation end product, which for lactic acid fermentation will be lactic acid. Why do you think, like our muscle cells, when we're fermenting glucose into lactic acid, why do you think that when the muscle cell generates NADH by glycolysis, it doesn't keep the NADH around for aerobic respiration, where we could make a lot more ATP from that, from aerobic respiration, than we could from fermentation, which actually the NADH is used in the fermentation step, but you make no ATP out of that. Any questions? Any answer there? I'll give you a point if you get it right.
So wait, what are you asking?
I'm saying why, when our muscle cells, for example, which can engage in fermentation, converting glucose into lactic acid, why does the muscle cell not retain this NADH and simply wait until oxygen comes to the muscle and then use that NADH in electron transport chain to generate more ATP than it could in fermentation, which doesn't generate any ATP? Why does the muscle cell not keep that NADH around and then use it in aerobic respiration? Instead, it uses it in fermentation to break
Well, it
down.
needs
Huh?
it needs NADH to keep the fermentation cycle going.
Yes, it needs NAD plus being
Yeah,
generated here because we need NAD plus in um, glycolysis. And if you don't have any NAD plus, you cannot engage in fermentation. And if you engage in no fermentation, you're not going to make any ATP because the muscle cell will only engage in fermentation when oxygen is not present.
yeah.
Okay. And the muscle cell will want ATP so it can continue working. And so it does that by engaging in fermentation. So send me your email and I'll get you the extra credit point. Okay?
On Canvas.
Yeah, send it to me in email and Canvas because I won't remember. I'm not even sure who did because I'm not seeing who talked. I'm only seeing my screen because I've got my
Oh,
screen up. Okay?
oh, I'm
Just
Alexandra, by the way.
okay, just send me an email. I'll make sure you get your extra credit point. Okay? All right. Um anyways, we talked about a muscle cell can engage in fermentation. It only does this when the muscle cell has oxygen limited, meaning the muscle is running out of oxygen. And the reason why it's run out of oxygen is it used all of the oxygen by aerobic respiration to make ATP, but it still has some glucose around, no oxygen. And rather than the muscle not working because it needs ATP to work, it will fall back on fermentation. It'll take glucose, make two pyruvic acids, change the pyruvic acid in the fermentation step to make lactic acid. That's just one step. And that will regenerate the NAD plus. And then we can continue with the fermentation reaction, meaning continue with the glycolysis. So fermentation can continue, and the cell will engage in fermentation for as long as it still has glucose. So as long as the cell has glucose, even in the absence of oxygen, the cell will engage in fermentation. We do generate some ATP right there, and then the muscle cell can continue working. Usually in every Olympics, I don't know if it's happened this year or not, there's one Olympic athlete who uh, pushes himself to the limit and actually has no more glucose in their muscle cells. They can no longer generate ATP, so their muscle cells cannot work. That athlete just collapses to the ground. The coach runs out in the wintertime, throws a blanket over the player, and then the player gets up after, I don't know, a little while, and then crosses the finish line, usually walking across. Anyone ever see that? It usually happens one time in each Olympics. In the summertime, I think the coach goes out and sprays the player with water, and then the player gets up and crosses the finish line, usually walking. Anyone ever see that? Usually happens one time in the Olympics. What happened was the Olympic athlete used up all the glucose in their muscle cells. There's no more glucose, no more ATP. Once they use the last ATP, the muscle cells can't work and their muscles stop working, they collapse. They're fine. Their heart and the lungs are working like mad to supply oxygen to the muscle as well as glucose to the muscle. Once the glucose and the oxygen come to the muscle cells, the cells can then work by aerobic respiration. They generate ATP and then the muscle can work, and the Olympic athlete stands up and then usually walks across the finish line. I'm sure you've all seen that. Like I said, it happens usually once every Olympics. I don't know if it's happened this time, but it usually happens once in the Olympics.
Normally, when the body runs out of glucose, though, it switches to fat. for fuel.
No, I'm talking about the muscle cell itself ran out of glucose. The heart and the
Oh,
lungs are working like mad to bring more oxygen and more glucose to the muscle cell. But if the muscle cell uses up all the glucose, no ATP is made, and then the muscle cannot work any further. And then the athlete just collapses. Okay. I'm not saying that the body has no glucose because um, most of these athletes have glucose stored in their liver and they're releasing the glycogen into the blood. It's just the blood and the heart and the lungs cannot deliver enough blood to the muscles to continuously feed the muscles with oxygen and with glucose. And all the time, all of us, we run out of oxygen in the muscle, and that's why we engage in fermentation. And you can feel the lactic acid start building up in your muscle cell because you'll have a muscle burn from the lactic acid being burned. I'm sure you've all felt that happening, lactic acid burning up, and that's from fermentation generating lactic acid in our muscles. Most of us do not ever collapse because, like I said, to use up all of the glucose in your muscle cells, you really have to be pushing yourself to the limit. And usually in the Olympics, there's only one athlete that does that. Any question about any of that? All right. That's pretty much lactic acid fermentation. The muscles in our body, at least what is that called? The active muscles in your body can engage in fermentation. No other cell in our body can engage in lactic acid fermentation. All of their ATP has to be generated by aerobic respiration. The muscles, though, get it both from aerobic respiration, especially when your muscles aren't working much, but when they're working heavily, you will fall back on lactic acid fermentation. Another fermentation reaction, which is a little more complex, lactic acid fermentation is just one chemical step, converting pyruvic acid into lactic acid. Uh, Alcoholic fermentation is a two-step process, converting pyruvic acid into ethanol by using an intermediate. That intermediate is actually a two-carbon molecule, so we get CO2 generated by alcoholic fermentation. Ultimately, we end up with ethanol. Which when you're making wine, the ethanol or the beer is what you drink. In the beer, the reason why it's fizzy is you cap the bottle and then you ferment it inside the bottle, generating the CO2, which creates the fizz in the beer. If you were to, in the wine, you normally make it in an open barrel or open casket, the CO2 vents to the air. If you were to make the wine in a cap bottle, it would become fizzy, and we call that champagne, where you make the wine in a cap bottle. And then the CO2 is kept to make the champagne. Any question about any of that? All right, lactic acid fermentation or alcoholic fermentation are two common fermentation reactions. There are a handful of different fermentation reactions. You don't need to know any of the others other than lactic acid and alcoholic or ethanolic fermentation. Here's the different types of fermentation reaction. Many cells can engage in lactic acid fermentation, including our muscle cells, streptococcus and lactobacillus bacteria that can ferment milk into lactic acid to make our yogurt. Yeast, Saccharomyces cerevisiae, can engage in alcoholic fermentation to make ethanol and CO2. There are other fermentation reactions. You don't need to know the name of them, but actually propionic acid fermentation is how you make Swiss cheese. Swiss cheese makes propionic acid instead of lactic acid, why the cheese tastes different. And the CO2 being generated is why the Swiss cheese has a hole in it from the CO2 being generated. There's Clostridium, which can make butyric acid and a few other products. Escherichia and Salmonella, which can make ethanol and lactic acid. Enterobacter, which can make formic acid and a few other acids. One of these, I think it's this one here, is how we actually make vinegar. You take alcohol, or actually glucose, you make wine out of it. You then ferment the wine. The alcohol will be converted into the acid of vinegar, which I think I thought it was acetic acid, but maybe it's formic acid. Any questions about fermentation? Here are different fermentation end products. You don't need to know any of them except for ethanol and lactic acid. Some industrial uses, making beer, wine. It's also how you make sourdough bread. Lactic acid is very common. It's how most cheeses are made, like cheddar cheese. Yogurt's made that way, rye bread, sauerkraut, summer sausage, Swiss cheese using propionic acid fermentation. You don't need to know that. I'm just telling you that. All right, a good summary, summarizing aerobic respiration, anaerobic respiration, fermentation. In aerobic respiration, oxygen is the final electron acceptor. We generate 36 to 38 ATP. For prokaryotes, it's always 38. You don't need to know that column. For anaerobic respiration, it's the final electron acceptor is a molecule other than oxygen. The generation of ATP is always fewer than 38 and more than 2. So something between 4 and 36, depending on which form of anaerobic respiration it is. And fermentation can happen either in aerobic or anaerobic conditions. We only generate 2 ATP, which doesn't happen in the fermentation steps of fermentation. It happens in the glycolysis step of fermentation. And what the final electron acceptor here is an organic molecule, usually pyruvic acid. Pyruvate usually is the one accepting the electron. Any question about any of that? All right, apart from glucose and carbohydrate metabolism used as food to obtain energy, we can use lipids and proteins. You can use a carbohydrate itself, like when you eat a bunch of starch, you break the starch into glucose and then send glucose down aerobic respiration in the normal way, as I showed you. You can also eat a lipid diet or a protein diet and burn these two and generate ATP by catabolism. Most cells can use catabolism to generate ATP, including our cells. A problem with a large molecule like a lipid or a protein is they cannot be immediately digested by the gut because the lipid and the proteins are too large to cross the cell membrane. How we solve this is we secrete extracellular enzymes to digest the large molecules outside the cell, break them into their monomers, and then bring the monomer across the cell membrane to be used by the cell. Like a protein, you eat a high protein diet and your body wants to digest the protein, you'll secrete proteinases outside your cells in the gut, which will digest the protein into amino acids. The amino acids then be transported into the cell, usually by active transport proteins, to be used by the cell. The amino acids will, especially if you're using a lot of food or something to use your muscles like a marathon runner, they can burn protein, but it'll be converted into amino acid. And then the amino acid, at least for most of us couch potatoes, will be converted into glucose. And then glucose will be sent down aerobic respiration in the normal way. You can, in your marathon run, directly burn lipids, sending the lipids into aerobic respiration. But a marathon runner does not do that until 40 minutes into their marathon run. So for the first 40 minutes, they're burning glucose and other carbohydrates, converting the carbohydrate into glucose, and then generating ATP for the first 40 minutes of their marathon run. After 40 minutes, the lipids can be digested. The lipids will be broken into 2 carbon chunks. The 2 carbon chunks will be converted into acetyl coenzyme A and enter the Krebs cycle that way. So you can burn lipids if you're a marathon runner and you're using a lot of energy in your marathon run, but you have to be running for at least 45 minutes before you can directly burn lipids. Before that time, you will not burn lipids, you'll be burning glucose and other carbohydrates. And if you're a couch potato and you're eating a high fat diet, you won't burn the lipids directly. You convert the lipid into glucose and then send glucose through aerobic respiration the normal way. Any question about any of that? So to burn a lipid, you'll first convert it into glycerol and into fatty acids. The glycerol will go into the Krebs cycle midway through. The fatty acids will be converted into 2 carbon chunks, converted into acetyl coenzyme A, which will then enter the Krebs cycle. And that's how lipids will be used if you're digesting lipids and then burning them directly. Proteins will depend on which amino acid it is. The proteins will be digested extracellularly. The amino acids will be brought into the cell. Depending on what the amino acid is, some of them will be converted into pyruvic acid, entering aerobic respiration here. Others will be converted into acetyl coenzyme A, entering the Krebs cycle here. Others will be entering late in the Krebs cycle, entering there. And that's the way proteins can be digested, assuming you're burning the proteins directly. Most of us couch potato, if we're to eat a high protein diet, we will not burn the protein directly. We'll break the protein into amino acids. The amino acids will be converted into glucose, and then glucose will be a sugar, which will enter the aerobic respiration pathway in the normal way. Any question about any of that? The point is that the metabolic pathway will get the oxidation of glucose in aerobic respiration to generate ATP. It's the most efficient way to generate ATP. And like I said, glucose is the preferred molecule. If it's another carbohydrate, it'll just be broken down and converted into glucose and then sent down aerobic respiration in the normal way. For us couch potatoes, we'll convert that molecule, whatever we're eating, into glucose, and then glucose will be sent down in aerobic respiration in the normal way. But if you're a marathon runner or some Olympic athlete who's constantly using up ATP, you can burn the lipids and the proteins directly. But most of the time, even they, when they're not currently using their muscles, they will convert fats and lipids into glucose and then send glucose down aerobic respiration the normal way. ATP is needed by your cell for many different cellular processes, including active transport of the molecules across the cell membrane. ATP is used in movement and in the synthesis of new cellular components. All anabolic reactions occur because the energy supplied by the reaction comes from ATP. Most ATP used by a cell actually is used up in making a new cellular component in anabolism, including the biosynthesis of a larger molecule. All right, so I did mention that if you eat a high protein or high carbohydrate diet, we'll convert these proteins and carbohydrates in various steps. First, you break them into their monomers, and then you convert the monomers into glucose. How that happens is by common intermediates. And I've talked about anabolic and catabolic pathways as distinct and separate pathways. That's not the case. In reality, they are joined together. They do have common intermediates. We have an amphibolic pathway, and that means the metabolic pathway can move in both directions. We don't have, I don't know, come on. I'm sorry, my mouse isn't working. We don't have the reaction always happening A going into B. Sometimes B can be converted into A in an amphibolic pathway. These amphibolic pathways are involved in carbohydrate, lipid, and protein metabolism. And everywhere where we have an arrow, it means double arrow. It means that the reaction can go this way from malic acid to oxaloacetic acid, but as you can see, we can convert oxaloacetic acid into malic acid. And let's say you're eating a high protein diet and you're currently sitting on the couch watching TV, like most couch potatoes. You convert that amino acid into oxaloacetic acid, convert that into that, 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 convert that into citric acid, convert that into acetyl coenzyme A, convert acetyl coenzyme A into pyruvic acid, go all this way backwards in glycolysis and make glucose. When your cell then needs ATP, it will convert this glucose you just made from eating your protein, which will be converted into the amino acid, which will be converted into glucose. And then you send glucose down aerobic respiration in the normal way. That is the way most couch potatoes and most of us will digest the lipids and the proteins. We'll convert them into glucose, and then when the ATP is needed, glucose will be sent down aerobic respiration in the normal way. And here for a lipid, we have lipids broken down into glycerol and fatty acids. Fatty acids broken down into acetyl coenzyme A. And let's say the fatty acid will be converted into acetyl coenzyme A. We don't use acetyl coenzyme A to make ATP because you're a couch potato. You're not using up ATP at the moment. So you convert the acetyl coenzyme A into pyruvic acid, make it go backwards. And by the way, this is double arrowed because the arrow goes this way and that way. And so once we make this from this, we'll then go that to make that. And then you'll make this way to make glucose from your lipids. Any question about any of that? And by the way, you all know that when you eat too much sugar and carbohydrates, you'll break it down into glucose, you'll break it down into acetyl coenzyme A. If you don't need the energy, you won't use acetyl coenzyme A to send it through the Krebs cycle. You'll take the acetyl coenzyme A and convert it into a fatty acid, which will combine with glycerol to make a lipid. And then you'll start storing that lipid down in your body. And that's what's will happen if you eat too much carbohydrates. You convert it into lipid and then store it as a lipid on your body. Any question about any of that? That would be true too. Let me see, I don't have this shown here, but if you were to eat too much protein, you convert it into amino acid, which will combine into different places. Let's say this amino acid is converted to oxalic acid. You have plenty of ATP around, you don't need any more, so you convert the oxalic acid into this way, going backwards, convert it into acetyl coenzyme A. Let's say you already have enough glucose stored down as glycogen in your liver. You don't need to convert any more into glucose. So what we're going to do is we're going to convert this acetyl coenzyme A into fatty acid. You'll then convert the fatty acid, combine it with glycerol to make the lipid, and then store that lipid down in your body as fat. Any question about any of that? How we're doing time-wise? Yep, I'm a little past. Whenever I run past and I don't notice it, let me know and I'll stop right there. Any questions? All right. I'm going to give you a 10-minute break, so the lab will not begin until 7:35.
Do you mean 6:35?
Excuse me, 6:35. Yes, 6:35. All right. No questions. I'll say goodbye and I'll see you in the lab at 6:35.